Hi everyone, so today we're going to take a little bit of time to look at the lac operon. So this is something that we looked at when we were first introducing gene regulations and operons. So I'm just going to go through the idea behind the lac operon and how things are regulated, and a couple like mutations that you might want to think about and how that changes the levels of lac-Z or lac-Y in a cell. So to start off, the operon includes the lac-Z, lac-Y genes, which transcribe two things. So lac-Z is responsible for actually transcribing the enzyme, which will break down lactose. And then lac-Y is responsible for basically allowing lactose to enter the cell. Okay. Right, so then now in terms of the repressor protein, so the lac operon is controlled by a repressor protein. So what I mean by that is that operons generally have this operator region, which is just a sequence on the DNA that's recognized. And so the operator is recognized by a regulatory protein, which is a repressor or activator. So in this case, the lac-I, once it's transcribed and translated, it'll produce the lac-I repressor protein, and when lactose isn't present, it can bind the operator region, which is a sequence of the DNA, so it's recognizing it's basically protein interaction to DNA interaction, and it weakens this promoter. So it weakens promoter 2, so that when polymerase comes around and wants to transcribe lac-Z and lac-Y, it can't as well anymore because lac-I is bound there. So talking about a repressor, generally when operons use repressor proteins, it's because their promoters are normally very strong. So this promoter right here is quite a strong promoter because normally when the repressor isn't bound to the operator, the transcription levels are quite high. And so we need that repressor to sort of like tone things down. So you wouldn't generally see an operon that had a weak promoter using repressor because the levels are already very low to begin with. So repressing won't really change transcription much. So in this case, we see a strong promoter, and that's being repressed through an operator and a repressor protein interaction. So P2 is actually a very strong promoter, which is an important thing to consider when we're looking at certain aspects of like what happens after certain mutations. Okay, so then we have lactose that comes around. So when lactose is present in the cell, it can actually bind lac-I and prevent it from binding to the operator. So when lactose is present, it binds lac-I and it basically morphs it into a shape that can no longer bind the operator. And so basically you have no repressing of your operon, and so like things will continue as normal. So you have your polymerase binding to your promoter. It's a very strong promoter, so you'll have quite high levels of transcription of lac-Z and lac-Y. And I know this came up a few times, was how can lactose actually first enter the cell to inhibit lac-I if you need to make lac-Y to allow lactose to enter, right? That's what we said here. So just to remember that lac-I binding to the operator isn't 100%. It's not happening all the time. There are still some moments in time where polymerase can bind to the promoter, and that's called basal transcription. So there's always a basal level of transcription occurring when lac-I is inhibiting or is basically repressing the operon. So that's something to remember there, is that there's always basal levels of lac-Y being produced, so lactose can always enter the cell if it's present in the cell's environment. Okay, so that's sort of just like an overview of what happens. So we can think of a couple things that might occur. The first being, what if we actually mutate the operator? So if I remove the operator, Let's do this in red. So if I remove the operator, so there's some mutation in the operator that makes it so nothing can bind to it, basically. So it's just not functional anymore. So we can ask ourselves what would happen in this case. So normally the operator functions as like a landing pad for the repressor protein. So when lactose is absent, LAC-I would be able to bind the operator and decrease transcription of LAC-Z and LAC-Y. But in this case, no matter if there's lactose or not, LAC-I is unable to bind the operator because there's just no site for it to actually bind anymore. It doesn't recognize, it can't find like its sequence because it's not the same as it's used to seeing because it's been mutated. So in this case, in the absence of lactose, in the wild type, we would see repression of transcription. But in this mutated version, we don't see repression of transcription because nothing can bind to the operator. But in the presence of lactose, in the wild type, normally LAC-I is it's prevented from binding to the operator. So there's normal levels of transcription. But in the mutant, in the presence of lactose, it would bind to LAC-I and prevent it from binding to the operator, but there isn't an operator to bind, so it doesn't really matter if we're blocking LAC-I from binding to the operator or not. So in the case with lactose, you would see similar levels of transcription to the wild type, but in the case, in the absence of lactose, then it would actually matter. So hopefully that gives like an idea of how you would go about the questions with the mutations, and also like a general picture of how the LAC operon works.